Hello and welcome to part 2 of chapter 14 on gene expression. We're talking about the last two topics here, translation and mutations now. We'll start with translation. And here's a reminder of that central dogma of molecular biology going from DNA to RNA to protein. We've talked about the DNA to RNA transcription. Now we'll talk more about translation. Like these other processes, translation can be split into three phases. Initiation, it has to start. Elongation, that protein needs to be made, so it's elongating one amino acid at a time. And termination, it has to stop somewhere. And so let's look at that, but first let's look at some of the important players, the things that we need to do translation. So we need all these things shown here. So translation requires a piece of message. We have initiation, elongation, release factors. So these are just proteins that are important for starting, extending, and ending the process. The other thing we need are these tRNA. These are transfer RNAs. I also like to think of them as translating RNAs because these are the molecules that bridge that gap between the nucleic acid information and amino acid information. These proteins here, these amino acyl tRNA synthetases, these are the enzymes that make, or I should say they don't make the tRNAs, but they attach the amino acids to the correct tRNA. And then maybe most important of all is the ribosome. This is the protein building machine. But let's look at those tRNAs first. Those are so important in this translation. So we can look at tRNA structure in a variety of ways. It is made out of RNA. They're called tRNAs. And they have a polarity of 5' to 3'. There is base pairing, but we have a single strand here, and the base pairing is occurring kind of like hairpins forming here. And it forms this two-dimensional cloverleaf type structure. Here's closer to what it actually looks like, a little more almost L-shaped. Here's the cartoon version from your textbook, just sort of simplify the appearance of it. And there are a couple of other features of this to pay attention to. One is this anticodon. This is going to be what base pairs with our message. And then over on the other, the 3' end, is the amino acid attachment site. So this AAC, these AAC residues, this is where we have the amino acids attaching, and that's the 3' end. And so it's important that that anticodon matches up with whatever amino acid's going to go there. And we can see how this works. So here is a piece of message, 5' to 3', UCA, and this is a codon for serine. You could check it on your genetic code if you'd like. Now, here's a tRNA that's carrying a serine. Notice it's attached to this 3' end, the serine. But also notice this 5' to 3, got to follow this thing with 5' to 3'. This UGA, this anticodon, is also anti-parallel to the message, 5' to 3'. You get base pairing, so this is just complementary base pairing, and that's how we have this translating molecule. Again, it's transfer RNA, but I like to think of it as translating. All right, so here's a question for you. Now, keep that orientation in mind, the 5' to 3', and knowing that these have to be anti-parallel. So here's an RNA molecule, and here's a chunk of it here. We've got a couple of codons. And what I'm asking you here is, what would the anticodon loop be for the tRNA that will complement this codon? Again, keep that polarity in mind as you answer this. All right, so it would have to be anti-parallel to this. So if we went 5' to 3', going from right to left, it would be CGG. But given the choices, and as is written here, these are actually written the opposite way, 3' to 5'. And so that would be this anticodon here, GGC. Again, if we're reading it 5' to 3', CGG. And that codes for proline. So this codon, and you check your genetic code, that will be for proline. So of course, it's really important that the right tRNA with its anticodon gets the right amino acid attached to it. And these enzymes called synthetases, these are responsible for that attachment. So in this case here, we have this tyrosyl tRNA synthetase that will attach a tyrosine to this correct tRNA. And so it's very specific that it gets the right RNA and it gets the right amino acid, puts them together, and now we have our charged tRNA. So that's what we call it when we have the amino acid attached. It's charged. You think of it, it's like it's ready to go. It's ready now to contribute to that protein synthesis. Here's a semi-three-dimensional image of what this might look like given the actual structure of tRNA, and here we see that amino acid being attached to it. All right, so what would happen if this didn't go right? So here's a question for you. So we have a mutant bacterial cell that has a defective aminoacyl tRNA synthetase, and it attaches lysine to tRNAs that have the AAA anticodon, which should get a phenylalanine. So what's the consequence of this? All right, so yeah, I mean, basically any time that this codon is encountered, that a lysine will be added to the protein instead of phenylalanine. And that doesn't mean the protein won't contain phenylalanine at all. This is to the second point, because there are other codons that code for phenylalanine. But if they encounter this, the UUU codon, which would go with this anticodon, instead of phenylalanine, you'll get a lysine. All right, so here's another just critical player in this. And notice there's a theme here, right? There's an underlying theme of RNA, RNA, RNA. We need messenger RNA. We need transfer RNA. And with the ribosome, this is ribosomal RNA that we're using. So ribosomes are made out of protein and nucleic acid, RNA specifically, and it's the RNA itself that is catalytic. For a long time, because people thought all enzymes are proteins, sort of thought, well, this RNA is some sort of structural thing maybe. But no, it's the protein that's just there for structure. It's the RNA itself that's the enzyme. And remember, if you have a catalytic RNA, it's called a ribozyme. And so the ribosome is a ribozyme, and that's what's responsible for actually building those proteins, as we'll see. Looking at the structure of the ribosome, there's two parts to it. It has a large subunit, so we see in different views here, and a small subunit. And so it has these two components that come together when proteins are translated. Well, again, we'll see how that works. If we zoom in a little closer, take a cross section, if you will, of a ribosome, there are three regions within the ribosome. We call them sites, and they are the P, A, and E sites. And we can look at specifically what's happening in each of these, and we'll see this more dynamically once we see elongation happening when the protein's being built. But the A site is where new tRNAs are coming in. And those are going to bring their amino acids, so like we see over here in this image. The P site is the peptidyl site, so we have this growing chain. We have a tRNA with this growing amino acid chain attached to it. And the E site is the exit site, and this is where the spent, see it doesn't have an amino acid attached to it anymore, the spent tRNA is just ejected. It exits the ribosome. We can see this from more of a computer modeling standpoint and 3D structures. Here's our piece of message kind of draped over the small subunit here. Here are the A, P, and E sites of the ribosome. And here is this polypeptide chain growing out of the large subunit here. And it actually goes through what amounts to a pore in the large subunit. And this is the exit tunnel. And so this is where we see our new protein emerging. So those are all the players here. Let's look at how it works. So we have to start the process, right? So that's initiation. It has to start somewhere. What we have happen is the small subunit of the ribosome binds to the piece of message. So upstream from a start codon, there's a region of RNA that will be recognized by the ribosome and the small subunit of the ribosome. It will bind. And we also need this tRNA met. So it's actually a special initiator tRNA. Remember, every protein is going to start with methionine. And it will have this anticodon CAU, or if you read it the other way, UAC. But it's complementary to this AUG start codon. So you have to have that start codon. So these guys come together. We need a bit of energy. It's in the form of GTP instead of ATP. And that's what provides that fuel for this initiation. In comes here the large subunit. Now we have a complete initiation complex formed. The whole thing, everything is there, all the things that we need to start this. We can look in a little more detail here, noticing that there are some other proteins involved. So these are called initiation factors. And they're just, again, important for binding to the starting point on the message. So the small subunit knows where to bind. And in here comes this tRNA. And again, it will bind to the start codon. Those initiation factors, we don't need them anymore. And now in comes the next tRNA with the anticodon complementary to this next codon coming in at this A site. And that feeds us right into elongation. And so we can look at this in a cyclical manner. So here we have at the P site this could be our initiator tRNA, or in this case, we're seeing it already has a few amino acids attached. So this is a little further down the message. And here comes a new tRNA, binds to the A site. Here's where the ribosome does its work. So the ribosome, remember, it's this catalytic piece of RNA. It will transfer this growing chain of amino acids to the new amino acid. So that now the whole thing is one longer. Next, the ribosome will translocate, meaning it shifts down one here. So we see the new chain is now attached over here to this tRNA that just came in. The ribosome translocates, so it just shifts down by one. This tRNA that has just been, the chain has been taken off of, now is out of the ribosome, it leaves the exit site. We have this next tRNA now in the P site, and boom, we just cycle back around. A new tRNA can come in. So round and round we go, and that's how proteins are made. Let's look a little closer at that transfer of that chain, or here in this case, a single amino acid to the next one. So we have the P site on the left, the A site on the right here. So this is our new one with bringing this blue amino acid. Here's the one that was already there, the green amino acid in this case. And the ribosome catalyzes this peptide bond formation. We see that here. And it's specifically, this is called peptidyl transferase activity, which again sounds like a lot, but think peptide and it's transferring it to the next amino acid. That's all that's really happening. And again, what is catalyzing it? The ribosome itself, the RNA that's catalyzing it. Again, just another way to view this, where we can see the exit tunnel in this growing chain of amino acids. I have a few different images here just so one might resonate more with you than others. All right, so ribozymes catalyze what chemical reactions? All right, so all of those choices were correct because if we go back here, remember we've got a covalent bond, it is a peptide bond, and it is a bond between amino acids. And that's what proteins are made of. All right, so we've made our protein now, or polypeptide. Where does the message stop? Well, we already know about the genetic code, and we know there are 3 stop codons. So maybe the question is, what happens when the ribosome reaches one of those stop codons? There is no tRNA for those stop codons. So there's nothing to come in there and deliver the next amino acid, if you will. What will attach at a stop codon is something called a release factor. So we see that in this case as this yellow protein coming in and binding to a stop codon. And this is basically a signal for the ribosome to stop. And so off goes our polypeptide. It's cleaved off of this tRNA, and here the tRNA is kicked out. Now we have this free polypeptide. It's done. And then it does take a little bit of energy here. It doesn't just sort of just drift apart. Some energy actually has to be invested, and then this ribosome complex will dissociate. The large and small subunits split. There goes our message. Maybe to degrade, or maybe there's other ribosomes already attached to it and they're translating it as well. And that is how termination occurs. So what happens to that polypeptide? We already know a little bit about protein structure back from many chapters ago. Proteins, they'll fold and form that final structure. Sometimes many polypeptides come together to form the final protein. That's this quaternary structure made out of multiple polypeptides. But there's other things that can happen to these as well. So that folding is critical, but they may also be modified in many different ways. And this is really important because you could actually end up with sort of different functions out of even a single polypeptide, because it's modified one way in this cell and it's modified in another way in a different cell, and that's going to determine potentially a different function. And so I like this little figure here. This demonstrates some of the possibilities that can happen to produce your final target protein. And so here are some of the things that we can see. So let's see, let's pick a good one here, just simple one. Methylation. So just adding some methyl groups in certain places. That's important for some proteins to finish them off. Let's see, proteolysis. This is cutting a protein. So maybe a polypeptide is produced, but for it to be functional, it actually has to be cut into 3 different pieces. And those go to maybe different things, or then they come back together in a different way. This is actually also a way to break down proteins when we're done with them. So there's all kinds of chemical modifications that can happen to proteins. Now think about this from a functional diversity perspective. Let's take a genome, say human genome. There's say 25,000 genes. From any gene, you could potentially produce some different transcripts. Remember that alternate splicing that we talked about? So let's say you could produce 4 different transcripts on average from any given gene. Some are only going to be 1, some may be more, but you get the idea. So you can get more than one kind of transcript out of a gene. And all of the genes are called the genome. All of the transcripts are called the transcriptome. And then all of the proteins are the proteome, because you can get an individual polypeptide out of any one of these transcripts, and then think about all those potential modifications that could happen to those proteins afterwards. So you can get way more proteins than there are genes. So if you had 20,000, in this case, you could get over 1 million different proteins based on all these modifications that could happen afterwards. Another important thing when it might need to happen when proteins are being built is that they get where they're supposed to go. And remember back to when we talked about cell structure, we have 2 populations of ribosomes. Some ribosomes are free in the cytoplasm, and some are attached to the endoplasmic reticulum, the rough endoplasmic reticulum specifically. Now those ribosomes aren't permanently there. They actually will come and attach to the ER, and protein translation begins in the cytoplasm initially, and then a protein that has the appropriate signal, as we'll see, will get moved to the rough endoplasmic reticulum. And so what are those proteins? Think back to when we looked at movement of materials around cells, things that might become part of the cell membrane, proteins that are going to be secreted from the cell. Those need to be processed at the rough ER. All right, so how does that work? So polypeptide synthesis begins in the cytoplasm, as I mentioned already, and it will finish there if that's so ultimately where that protein needs to be. Or what really is happening is that protein doesn't have this what's called a signal peptide. So the very first part of this protein that's being produced, it has a certain sequence of amino acids that acts as a signal for, hey, you need to move to the endoplasmic reticulum. And so that's what we see in this cartoon here. This ribosome and messenger RNA complex is producing a protein here that's coming out as red. And if this signal peptide is present, this protein here, this is the signal recognition particle. So it's a protein that will recognize this, it'll attach to it. And that will basically bring or facilitate the docking of this ribosome to the endoplasmic reticulum. And here this protein now is being fed into the lumen of the endoplasmic reticulum as it's being produced. And so this is ultimately where this is going to go, as either in the cell membrane or be secreted. And it has to happen at the ER for that to work. Here we see this same thing again, just focusing on that SRP, that signal recognition particle. Here's the signal peptide. This SRP binds to it. That will allow attachment to the endoplasmic reticulum. The protein's produced, and then here is that finished protein floating in the endoplasmic reticulum. As we know, some of them are actually produced and embedded initially in that ER membrane. Okay, so this came up back when we talked about prokaryotes, this idea that multiple ribosomes can attach to a message. And we see this also in eukaryotes. It's just that the message needs to be modified first in eukaryotes, whereas in prokaryotes, as soon as that message is being produced, boom, 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 ribosomes are attaching to it. But in eukaryotes, the message needs to be modified, moved out to the cytosol, and then ribosomes can attach. But either way, we see this in both groups of organisms. And this is called a polyribosome. And it's an efficient way of translating this message, right? It's not waiting till, okay, we're completely done one protein, let's start again. No, multiple ribosomes can be making the same thing. So this is a great way to crank out that protein until, say, the message breaks down. So let's have a question here on translation before we move on to mutations. We've got a protein that's an important signaling molecule. It needs to be secreted. What has to happen before this can be secreted by the cell? All right, yeah, so it needs to have that signal sequence, and then that signal recognition particle protein will attach to the signal sequence. The protein will be fed into the ER, which then gets processed by the Golgi and then out to the cell, outside of the cell. All right, let's talk about mutation now. Now we've heard about our chromosomal, big scale chromosomal alterations, but now we're zooming into a smaller scale. And a mutation in general is a change in the genetic material of a cell. And we're going to first focus on point mutations, and that's an alteration of a single nucleic acid base. And we'll use sickle cell anemia as our example to start. We've already heard about this one throughout the course, and we'll hear about it again. And the disease is sickle cell anemia, and it's caused by this variant allele of hemoglobin molecule. And so here's our normal hemoglobin sequence. And here is an abnormal sequence. The difference here is one nucleic acid difference. We're seeing, in this case, just the DNA sequence, but you could convert that to RNA if you wanted. And that results in a change of a glutamic acid to a valine, or a difference, let's say that. That's a better way to describe it. This allele will produce hemoglobin with a valine at this spot. This hemoglobin will produce a hemoglobin with a glutamic acid at that spot. Now, the impact is huge because this one amino acid change results in the ability of hemoglobin to form these stacks, these chains, which doesn't normally happen, but it does here. And that will result in this cell sickling like we see in this image. Now, as we talked about before, if you're homozygous for this, that could be fatal at a young age without any kind of treatment. But if you're heterozygous, this actually provides some protection from infection with malaria. That's a parasite that gets into your red blood cells and lives and divides there. So another thing I'll point out is this might catch your eye. The difference between these two figures, this says GAG, for example, and this says GTG. And the example here shows a different nucleotide stretch, which is GAA and GDA or GUA. Both of those codons code for glutamic acid and valine, so the example still stands. But I will point out there are actually a few different mutations that can lead to sickle cell anemia that we see in the human population. Either way, though, it's that one nucleic acid change that makes a huge impact. Another really important point, and which sometimes gets confused when you're learning about this mutation didn't just happen. It's not that someone is born and then when they're 2 years old, a mutation happens and I have sickle cell anemia. This mutation occurred thousands of generations ago. The estimates are approximately 70 to 150,000 years ago in the human population. And really that has left us with these alleles that are really variants of hemoglobin. So the mutation itself occurred a long, long time ago. So the sickle cell anemia allele example was a mutation that is a missense mutation. We saw a change in the amino acid sequence, and that's why it's called a missense. And this example here shows another example is this substitution of a T instead of a C, which results in the change in the code here from a GGC to a AGC, and that goes from glycine to serine. So it's changed the amino acid sequence. Now, if the amino acid is very similar in property to the one it's replaced, maybe that doesn't have a big impact. But we saw with the sickle cell mutation that can have a massive impact. So it depends where this occurs. In this example, it's occurring at the very end of this protein. Maybe that's going to have a minor impact. If it occurs in the middle somewhere, or an amino acid that's part of an active site of an enzyme, that could matter a lot. So it depends where it is and whether that's a functional part of the protein and so on. Another kind of mutation that is basically not noticed at the phenotypic level is called a silent mutation. And here we see, so here's our wild type sequence. Here we see a mutation, A instead of G, change in the mRNA code here. So GGC to GGU. All right, there's a change. But look, glycine, glycine. Because remember, our genetic code is degenerate. There's more than one codon that can code for glycine. And both of these do. So we have had a mutation that's occurred, but you don't see it at the phenotypic level. That protein is still the same protein, and whatever it does is going to do the same thing, whether this particular mutation happened or not. Now, these mutations can be helpful if we're looking at the genotypic level. We can tell apart different alleles, even if they don't have a functional change. Another kind of point mutation is called a nonsense mutation. So here we, again, have a substitution type mutation, so adenine here instead of thymine in our wild type. But what's happened is a stop codon has been introduced. And in this little make-believe protein, this was only 4 amino acids long, but you've introduced a stop codon way earlier than where it should have occurred. And so this is potentially going to impact that protein dramatically if it occurs early in the sequence. If you introduce a stop codon, 2 amino acids from the end, maybe that won't matter. So it probably depends on where it occurs, but this is generally going to have a negative effect, right? Random chance probably means it occurs somewhere along the length of that protein, not down at the very end, and that protein will be shorter and it probably isn't going to function like it's supposed to. Now, keep in mind that this stuff is all the fuel for evolution too. So, okay, this protein doesn't have that old function anymore, but maybe a protein has a new function, right? So that could be important as well. So mutations aren't always all bad. They provide that fuel for natural selection. All right, another kind of mutation is called an indel. These are insertions and deletions. Now, the reason we lump them together is because when we're looking retrospectively at, whether this was an insertion or deletion, we sometimes can't tell. So here's an example to explain that. So on the right here is what we have, say, today. So we're looking at two different alleles, and we see, okay, here's allele 1 and 2, for example. We've got this what could be an insertion or a deletion. In this scenario, the top one, the ancestral sequence had this red region, and it was a deletion that resulted in allele A. And allele B down here is the same or similar to the ancestral sequence. Another possibility would be, well, the ancestral sequence didn't have that red region, and there was an insertion that occurred. And then this allele A is more like the ancestral, allele B had the insertion. So we often can't tell. We have the scenario on the right here where we have these two different alleles, but we don't know what had happened in the past. Was this an insertion or a deletion? All right, so let's look at the impact of these kinds of mutations. So here's an indel, a single base insertion. So this is an insertion. And we add an extra adenine here. Well, what's happened is it actually has introduced a stop codon. So that's bad. It's completely messed up this protein. It will cut it short. And so these kinds of events can have a dramatic impact. Here's a different example. In this case, we have a deletion. And we've removed this adenine, therefore here uracil in our code. And what's happened, our protein used to be just these 4 amino acids long, ending with this stop codon. But because we've taken this one base out, we've shifted that whole message over. Now we've actually lost the stop codon, and this protein will continue to be made until maybe it encounters another stop codon by random chance. And so this is going to have a massive effect. This is supposed to be 4 amino acids long. Now it's who knows how much longer. And in both of these cases, what we've done is caused a frame shift. Remember that start codon sets the frame, like where to start, and then we're going to count by 3s. Every 3, we're going to say this is an amino acid. But with this kind of mutation, it shifted the whole rest of this message just over 1 notch and completely changed it from that point forward. Now, if you were to have some mutation that removed 3 bases at a time, and in a long stretch, that might be not necessarily 3 consecutive, but 3 that are, somewhere along the way, you end up restoring at least parts of that amino acid sequence. So in this case, we've just taken out a whole 3 here, and we've lost this lysine, but that's the only change. The other amino acids are still there. Now, the odds of 3 in a row like this occurring at random is incredibly remote. If you've got this many mutations occurring, there's probably others that are happening elsewhere that are going to mess with this message. And so in this particular example, maybe this doesn't have a huge effect, but again, in reality, the random nature of mutations, these kinds of things are probably going to have bigger impacts. All right, so we've got DNA coding for an albumin gene, and there is a mutation that occurs. It's a substitution mutation, so we're swapping out a T and getting an adenine. This changes the RNA codon. So what kind of mutation is this? You'll have to check your genetic code. Okay, so yeah, this is a missense mutation because we've gone from UCU, that codes for serine, to ACU, which codes for threonine. So that's changed the amino acid that was coded for at that spot. So let's finish up with a couple of points here. One is that, mutations do occur. Sometimes they don't have a big impact like the silent mutations. And then even those that can maybe change an amino acid maybe that's not going to have a big effect. Again, sometimes they can. And so we kind of go from the sort of movie version of mutations where, a mutation's occurred and now this character has superpowers, versus the reality where if it's not a lethal or dangerous mutation, this codon change in this individual results in this glucose 6-phosphate dehydrogenase producing a little more 6-phosphoglucono delta lactone, right? So not as exciting in real life. Now, it might be helpful to think about these mutations almost like a language. So we think about a silent mutation here in this example, the cat ate the rat. If we sub out the C for a K, it's silent because the meaning is still maintained here. So we say the cat ate the rat, it still sounds the same. A substitution, this is where we have the missense mutation, where the sentence is changed. So now we have the hat ate the rat. It doesn't quite make sense. Deletions and insertions, we say add a letter in here. Now we have the ka, tet, her. Everything is kind of got messed up, and the same deal with this insertion. So we had, a deletion here, insertion here. So that's caused a frame shift in the message. Now, it's also important to recognize that some of these changes could be negative, but they are also the fuel for evolution, right? So where do new proteins come from? So sometimes proteins will become non-functional. Sometimes mutations will occur that will be beneficial. Sometimes mutations will occur that don't have any benefit in that moment. And so we can take an example of words. So we've got our protein with these 4 amino acids, GONE. So spells the word gone, right? And so we can have all kinds of mutations that occur. If we look, many of these changes account for nothing. Like, these are words that don't make sense. But sometimes changes occur that actually make a new and functional word. So we've gone from gone here to none, or bone, or done, or gene. So some changes can produce something different and useful. But again, many of these mutations don't amount to anything or maybe have negative impacts. All right, that's it for this chapter. We'll see you next time.